Started?
Recording in progress. Yes.
Okay, welcome everybody to Pediatric Grand Rounds. I'm going to have our Vice Chair for for Education, Dr. Sandrain Van Schaik, come up and introduce our speakers for today.
Welcome everybody to Pediatric Grand Rounds, both here in the live audience at the Obendorf Auditorium, where, as every week, we have delicious lunch, and to everybody who's joining us over Zoom. Today's a very special Pediatric Grand Rounds. It's a combined pediatric and bioethics Grand Rounds. And I want to give a shout out to Dr. Vatlaukes, who actually, in the end, couldn't be here today, but she was the person who came up with this idea and got this wonderful group of people together. It's going to be a multi-people group presentation followed by a panel discussion. And I will just briefly introduce both the speakers and the moderator to you. Let me start by with the moderator. It's Dr. Julia Brown. Dr. Julia Brown is an anthropologist and postdoctoral fellow in the UCSF Bioethics Program and the UC Berkeley Kavli Center for Ethics, Science, and the Public. She's also affiliated with the Center for Maternal Fetal Precision Medicine at UCSF and the Center for Global Mental Health at University of California, San Diego. She's author of the Clozapine Clinic, Health Agency in High-Risk Conditions, and she's currently conducting an embedded ethics ethnography on the emergence of prenatal gene therapy and the tensions around social inclusion for underrepresented communities. Her broad research interests are the ethics of controversial biotechnology, health equity, and lived experience of health and caregiving, and she will be the moderator of the panel discussion. And then we have three speakers that will start us off. I believe the first person to speak is Dr. Marcia Treadwell, who is a professor in residence of pediatrics at UCSF, and really has a lot of roles, actually. She's also the co-director of the UCSF Sickle Cell Center of Excellence and co-chair of the UCSF Benioff Children's Hospital Diversity, Equity, Inclusion, and Anti-Racism Council. She is a health services researcher and clinical psychologist and leads an internationally recognized program of research focused primarily on populations affected by sickle cell disease. She has led multi-institutional, national, and international interdisciplinary teams conducting research on how social determinants, psychological and behavioral factors, and organizational structures and processes impact the health and well-being of individuals living with sickle cell disease and their families. She leads one of 8 United States Sickle Cell Disease Implementation Consortium sites, which is funded by the National Heart, Lung, and Blood Institute. And she is a co-PI and regional director for a Pacific Sickle Cell Regional Collaborative, a 13 consortium state consortium that seeks to improve access to knowledgeable care for individuals with sickle cell disease of all ages. So if you are not impressed yet with the fact that she knows a lot about sickle cell and all the factors that play into it, there you go. The next presenter is Dr. Manar Zaglula, who holds a BS in biology and chemistry from the George Washington University and a PhD in translational biology. This is every grand round.
6 for adult acute care. Code blue A661.
And I've learned that talking over it doesn't help.
For adult acute care. Code blue A661. Hospital 6 for adult acute care. Code blue A651. 6 for adult acute care.
All right. So Dr. Zaglula's research background is in neurogenetics and genetic screens. He serves as the policy and engagement manager for the Innovative Genomics Institute. And prior to joining that institute, he was a science and technology policy fellow with the California Council on Science and Technology. In his dual role as a policy consultant in the Assembly Environmental Safety and Toxic Materials Committee and a legislative staffer to State Assembly member Dr. Bill Quirk, He analyzed environmental policy proposals, shepherded new bills through the legislative process, staffed a number of issue areas, including health, education, and housing, and engaged extensively with stakeholders and lobbyists. He's passionate about working at the nexus of science and policy and contributing to the translation of scientific advances to tangible benefits to humanity and the planet. And I'm really excited to have someone with such a different lens on things join this panel today. And then the last speaker is Christelle Grace Salomon. She's a Haitian American Boston native sickle cell warrior who found her home in California 6 years ago. Christelle has been living with sickle cell for 28 years until she received a stem cell transplant in January of 2022. Christelle's experiences as a patient play a pivotal role in her career goals as an inspiring physician scientist. When she's fully recovered from the transplant, Christelle plans to return to medical school to finish her training. In her free time, she enjoys catching rays on the beach, cooking dishes from old cuisines, and dancing kizomba and salsa. I do not believe that she will do any dancing here today, but we're so pleased to get yet another completely different perspective on this panel today. So with that, I'm going to hand it over to Dr. Treadwell. Dr. Treadwell.
Thank you so much. Can everyone hear me okay? Good. It's great to be here and to lead off this wonderful panel. I'm really excited. So we are going to be focusing on ethics and access for sickle cell gene therapies. If you're interested in related mechanisms and outcomes, you should review the grand rounds that Dr. Walters did March of 2022, where he talked about the curative therapies for hemoglobin disorders. These are our disclosures, and they don't have an impact on what we're going to be talking about. And our objectives are first to provide you with information so that you can articulate the unique challenges of introducing gene therapy in a disease like sickle cell disease that is so characterized by inequities. Next, we're going to describe access barriers to sickle cell gene therapies and how this might impact patient care. And then lastly, leave you with an appreciation, we hope, of the challenges of discussing curative therapies with patients. So sickle cell disease is well suited as a target for gene therapy. It's the result of homozygous and compound heterozygote inheritance of a mutation of the beta globin gene. So you have a single base pair point mutation that results in the substitute of valine for a glutamic acid in the 6th position of the beta chain of the hemoglobin. Red blood cells that contain hemoglobin S undergo polymerization and become rigid and sticky when exposed to deoxygenated environment. And the rigid red blood cells are subject to hemolysis and due to increased density affect blood flow and vessel wall integrity. And this leads to vasoconstriction, tissue ischemia, and infarction. So this cascade of events on a molecular level leads to a very serious and complex disease. And you can see that anywhere that blood flows, you can have an issue with sickle cell disease. And these acute and chronic complications really impact quality of life as well as quantity of life. And then further, sickle cell disease in the US primarily affects Black and Brown people, and they have been made vulnerable by systems to negative impacts of social determinants of health. And so this includes poor access to quality education, limited access to basic health care, food insecurity, housing insecurity, and limited access to intergenerational wealth. You might have seen this slide before. This is from the CDC, and it points out how health outcomes and health resources have really been characterized as by inequities and disparities for the sickle cell population. So the average life expectancy for people with sickle cell disease is a full 30 years shorter than people who don't have sickle cell disease in the US. And patients with sickle cell disease have the highest rate of returning to the hospital compared with any other condition in the US. Their rate of things like stroke is higher, as well as pain, infection. And then in terms of health resource disparities that impact ability to obtain healthcare and services. Many people are on public insurance, and this may limit their access to providers. It's well documented that healthcare providers inaccurately perceive patients with sickle cell disease as drug seekers and doubt the severity of their pain. And as a result, it's been shown that patients with sickle cell disease experience longer wait times to see a physician and get pain medication when visiting the emergency department, among other health disparities. And then lastly, particularly for adults, the number of physicians trained and willing to treat people with sickle cell disease is limited. And you also probably saw this last year with Dr. Walters. But we want to point out how sickle cell disease depicted in the blue, again, affects 100,000 people and affects primarily Black and brown people. And cystic fibrosis in the orange affects about 30,000, primarily white people in the US. You see an immediate problem here because with NIH funding going more so to cystic fibrosis out of proportion to their numbers in the population. And then the comparison is even more stark with foundation funding. So there's an enrichment for the cystic fibrosis population compared with the sickle cell population about tenfold. And this has a devastating impact on discovery and implementation. So the table shows you some detail that you'll see in this figure. The blue, in this case, is cystic fibrosis, and you can see a significant number of disease-specific PubMed listings compared with sickle cell disease. So there's a big disparity here, and this gap doesn't look close to closing. Then you have the number of FDA-approved drugs over time. And so in cystic fibrosis, you have a number of drugs that were approved in the time period between maybe about 1990 to 2018. In sickle cell disease, there were only 2 in that time period. So the access to disease-modifying therapies is extremely limited, has been for people with sickle cell disease. Okay, so next we're going to turn to Dr. Zagulula is going to be talking about costs.
Right. Can you all hear me well? Great. Good afternoon, and thank you for inviting me to this excellent panel. Really thrilled to be here and representing the Innovative Genomics Institute. For those of you who don't know us, we're actually a partnership between UCSF and UC Berkeley, founded by Jennifer Doudna, and our mission really is to bridge genome editing technologies to affordable and accessible solutions in human healthcare, but also in climate and agriculture. Before talking about access and affordability issues, I really want to start off by introducing you to some of the transformative, potentially even curative technologies that are coming to market for a sickle cell. So traditionally, when we talk about gene therapy, we really mean gene addition therapy. And this is a process by which a virus encodes a gene of interest that is then introduced into the target cell. And just 2 weeks ago, Bluebird Bio introduced lovo-cel to the FDA for a biologics license application, meaning they've done all the clinical trials and they now want approval of this drug. And what lovo-cel does is it introduces an anti-sickling hemoglobin into the hematopoietic stem cell. And as Dr. Treadwell mentioned at the top, sickle cell is caused by point mutations. So if you're able, research has shown that if you're able to replace 20 to 25% of the cells with the sickle cell mutation with normal copies, you actually get a functional cure in patients. Another approach to this problem is using CRISPR. So CRISPR-Cas9 is a bacterially derived precision genome editing tool that has the ability to either knock out a gene, so disrupt the genetic function, or insert new DNA, or repair specific point mutations. And so in this case, Vertex Pharmaceuticals and CRISPR Therapeutics teamed up to generate exa-cel. And exa-cel actually targets a suppressive genomic region of the fetal globin gene. So fetal globin or gamma globin can replace beta globin in the production of hemoglobin and reduce the number of sickling cells. And both lovo-cel and exa-cel have shown dramatic improvements in clinical trials. So for lovo-cel, there was a 90% or 90% of all trial participants actually lived without any vaso-occlusive events for 6 to 18 months after infusion, which is honestly miraculous. And then another CRISPR approach would be a CRISPR repair approach. This is a little bit earlier in trials. So an IND or an investigational new drug application has been submitted to the FDA by a collaborative team between UCSF Benioff, UCLA, and the IGI. And so what this does is it actually introduces a DNA template for repair of the hematopoietic stem cells DNA. So rather than disrupting something that is not specifically the sickle cell genetic variant, it actually introduces a repair template that the cell can use in a process called homology-directed repair after Cas9 cuts the DNA at precisely the spot where the mutation in the sickle cell mutation occurs. Now, as you can see here, the price tags of these genetic therapies are already surpassing $3 million, which is a struggle for private payers, let alone state Medicaid programs, which have limited budgets. And it's a huge question how outside of industrialized nations, patients in low and middle income countries would ever be able to access these, even if pharmaceutical companies negotiate prices that are lower in outside of the U.S. This still is a huge issue and will be moving forward, especially as more and more genetic therapies are coming to market. Now, the price tags of these drugs are high not only because for-profit pharmaceutical companies have a shareholder responsibility to maximize profit, but because the R&D or research and development pipeline is actually very costly and lengthy. And so it requires extensive preclinical testing, phase 1 through 3 clinical trials to demonstrate safety and efficacy of a drug, extensive quality testing along the way, and then lengthy post-market monitoring periods as well. And so traditionally, government grants and philanthropic dollars have covered discovery, preclinical trials, and sometimes into phase 1-2 trials. But really at that stage, for-profit pharmaceutical companies and biotech companies that are backed by venture capital firms have to come in and buy or sponsor a program to bring it, to shepherd it through phase 3 trials and onto the market and then into the post-market monitoring period. And that's really where a lot of the cost racks up and why we see such high price tags. And so to illustrate the point of why, in the current model at least, we need private industry to come in and take over from the academics and from nonprofit pharmaceuticals, I want to highlight some of the cost pressures along the R&D pipeline. So the median capitalized R&D investment for a single drug on average is $1.14 billion. And this is based on an analysis of 63 drugs between 2009 and 2018. And that very high price tag or amount actually also includes failures, which are significant. So less than 10% of all drugs that start off in phase 1 trials actually make it to FDA approval, with attrition at every step of the way, but especially in phase 2 and phase 3 trials, at which point a lot of the costs have already racked up. Then for phase 3 trials, which sometimes are also referred to as pivotal trials, if you hear that term, they can have a very wide range of costs. So depending on drug product, patient population size, and trial design. The mean cost for a phase 3 trial that does have a control group is $35 million, but that could drop down to, say, $5 million for an extremely rare disease where a control or a placebo group would actually be unethical to be included, or it could be as high as $350 million for a non-inferiority study between 2 drugs where you have to have a very large patient population because the effect sizes you're trying to detect are so small. Manufacturing to FDA or other regulatory compliance standards is a huge cost pressure. So besides the skilled personnel that is required for these endeavors, the cost of goods is actually dramatic for biologics, and especially in the cell and gene therapy space. So, for example, Sarepta's gene therapy for Duchenne muscular dystrophy, which is currently under review by the FDA and actually has an important hearing tomorrow, the estimated cost for a single dose of that gene therapy is $100,000 just to make the compound. And then there's continuous quality testing and logistics and other cost pressures on the manufacturing and regulatory side as well. And then, as I had alluded to earlier, cost of capital is significant. So in the same study as in the first bullet point, the cost without capitalization would actually drop from $1.14 billion to $770 million. So that's about a third in cost savings if you didn't have to rely on the traditional venture capital with high ROI. And then lastly, post-approval, there is a lot of costs associated with maintaining the drug on the market. So marketing, physician education, continued quality testing of every lot that is released for the public or for the patients. And then the FDA recently came out with a guidance to recommend up to 15 years of post-market monitoring period, which is very substantial. And so the IGI, we've put together a task force led by my boss, Dr. Melinda Kliegman. And I want to punt this to the panel discussion because I'm running up on my time, but just to briefly introduce, so we've convened experts and talked about how do we design an alternative framework to the traditional for-profit venture capital model that is currently dominating the space. And we've talked about pricing and access issues of manufacturing and regulation, intellectual property and licensing, which is a huge component, and then innovative organizational and funding models. And I'll give it over to Christelle.
Dell and I are going to take the next section, but it is me again. So so we're going to actually, with that context of the health disparities related to sickle cell disease and the costs that were just gone over, we're going to talk to you about how this can impact access and then the ethics of sickle cell gene therapies. So first, I want to go over systematic review that was done by a team in the United Kingdom that really pointed out how patient and public perspectives on cell and gene therapies have been shaped over the course of the last few years. So what they determined was that sort of the main issues are just overall acceptance. Would you accept a cell and gene therapy? But you have to have some knowledge and understanding, and then an understanding of risks and benefits. And then where are you getting that information from? So with sickle cell disease, you have to really think about that trust in that relationship, where people are getting their information, what information they're getting is shaded by the decades of abuse and exploitation that the population has experienced, the neglect, and the significant underfunding that we've gone over. Next, I wanted to point out that the National Academies of Sciences, Engineering, and Medicine had convened a committee or panel recently, 2020, where they looked at specifically the impact of sickle cell disease on the population from a physiological, social, psychological standpoint. They really incorporated issues of racism and discrimination, and specifically pointed out what some issues are related to the technology of gene therapy. And so they highlighted that prioritizing an expensive technology when people don't have adequate access to comprehensive care is a true ethical dilemma. And so that you can't proceed with these expensive technologies without really designing and implementing patient-centered care that is very much lacking. So you need not only the care now, but because many people aren't eligible for the gene therapies, but you also need long-term comprehensive follow-up. And so a person-centric model needs to be implemented. Another issue, if you think about in general, informed consent and therapeutic misconceptions about what the trials are about and how they really are different or the same as treatments, misestimations, how much, again, have you heard about the technology and do you trust it? Do you believe that you're going to benefit from it? Do you downplay in your mind the risks and benefits? So without a really good understanding of an individual's conceptions and estimations of the risks and benefits, and colored by a history, again, of this discrimination and mistrust in the healthcare system that people with sickle cell disease rightly have, then you may end up with inadequate informed consent. And Christelle is going to be talking about how it really has to be an ongoing process that's dynamic in terms of informed consent. If you don't go through that, then it's really not respectful of people who are literally putting their lives on the line. And then in our pediatric setting, we want to think about minors in particular, in that assent is needed from mature minors. Their decision usually prevails, but studies have shown that their comprehension may be as low as 50% of things like what are the aims, what are the procedures, and so on. And there hasn't really been enough study in this area about strategies to improve comprehension. And then finally, we've been talking about sickle cell disease with a population of 100,000 people in the United States, but more than 300,000 babies are born every year with sickle cell disease in sub-Saharan Africa, and they aren't expected to live, many of them, beyond their 5th birthday. So I am also a member of the Ethical, Legal, and Social Implications Working Group for the Sickle in Africa Consortium. And the group has outlined what some of the issues are, again, in a global context. And Manar touched on that costs may limit equitable global access. But another point about that is that many people in African countries pay for healthcare out of pocket. And so they can barely pay for daily medications and treatments. And so something like gene therapy is way beyond their reach. Next, you have that there aren't as many legal and regulatory guidelines. So very few countries actually have guidelines about these technologies. And the public health infrastructure is bad in many countries, so that access to comprehensive sickle cell care, hydroxyurea, prophylactic penicillin, basic care may be very limited. And then lastly, again, what my colleagues pointed out to me that I hadn't actually thought about is that, we're doing these trials on young adults and adults, but when you have children who aren't going to live beyond their 5th birthday, then who should your population be that you're looking at in terms of the gene therapies? So I'm going to turn it over now to Christelle, who's going to talk to you about beyond those numbers and statistics.
I am rather short. Can you all see and hear me? So I thought it'd be really important for me to share my intersecting experiences as a patient with sickle cell disease, a former medical student, and a now transplant recipient to help contextualize and give a more tangible understanding of the data that's been presented so far. So my experience can be kind of split up between patient and student doctor, and there is a place where I think they intersect. And so as someone living with sickle cell disease, I face a lot of barriers to getting basic care that Dr. Todrell just talked about. And I remember living in SoCal, and that's where I did my grad school and part of medical school. And I faced insurmountable barriers to getting access to a sickle cell specialist. Out of the 3 years that I was living there, I had a sickle cell specialist for 5 months. And it was really hard getting access to medication like hydroxyurea and Adakveo and Oxbryta if I didn't have a sickle cell specialist. And Medi-Cal kept denying my referral to sickle cell specialists, but then I couldn't get the medication I needed because I didn't have a sickle cell specialist. And so I was in this constant loop of not being able to get past that barrier of, I just need something to help me modify the life that I have right now. And once I finally got access to a sickle cell specialist in my last 5 months, I thought that the issues I had with discussing novel therapies and different disease modifying medications and curative therapies. I thought I was beyond that, but it turns out that wasn't really the case. I still had a lot of difficulty accessing balanced and productive conversations about curative therapy. And I find that when I would bring it up, I was met with the, "You're not dying yet. Or your kidneys are mostly functioning fine, and you're at a 6 out of 10 pain, and last week you were at an 8. So like, we're doing better. You don't really need a curative therapy right now." And when I had approached my clinician to help, I didn't even want to go there. And I was like, can we think about this process as I embark on these current therapies that I'm taking? And I was met with very stark, just trying to convince me very, very, very deeply that I was in the wrong, even though I had a matched 12 out of 12 HLA donor, that I should not even consider pursuing this. And it was honestly me connecting with old coworkers, colleagues that I knew, watching videos online about gene therapy, about stem cell transplant. That's how I gathered my information. And before I went to med school, I wondered, how is it that every time I go to the hospital, I can't seem to get what I need? And it made sense after my hematology oncology block, when we had, I would probably count on my one hand how many slides we talked about sickle cell disease. There was one slide that mentioned in one short bullet point that there was pain. We didn't talk about anything else. We didn't talk about any other presenting symptoms. We didn't talk about the social medical issues that patients with sickle cell disease face in the hospital. We had many stigmatizing conversations about narcotic use. And so it makes sense, how can we expect clinicians to be prepared for a conversation about moving a patient's life forward when, as a system of medicine, we haven't prioritized education about that patient population, treatment of that patient population, and imagining a better life beyond everyday pain. And so that really put some things together, and lines up with what we've been talking about today. And another really important concept to think about when we talk about cures is the idea of identity. And in a paper by Orris et al., they talk about and do a qualitative study about the intersection between chronic illness and identity. And there are 4 different ways you can integrate your chronic illness into your identity. There's one that's engulfment, where your condition consumes your identity. There's one where you reject it, where you just don't associate with the condition that you have. And then there are 2 more adaptive mindsets, where you accept that you have a condition, and then the other where you develop a growth mindset and you are able to become enriched because of the challenges that you experience. And a lot of patients with sickle cell disease fall in this category. Sickle cell disease bleeds into every single part of your life, socially, professionally, mentally, physically. And so you really learn to adapt and live with your life as a sickle cell patient. And that's how I felt. And so now to think that this disease that I've lived with for all these years, I've shaped my community around the people who have sickle cell disease, my nurses and my doctors. And now if I don't have that, who am I? Am I still a sickle cell warrior now that I've received a transplant? And that is a really hard thing to grapple with as a person who spent your whole life living with this condition. And not to mention that some of the curative therapies that we talk about, if it's not quite changing your genotype, but you're phenotypically normal, do we call it a cure? Do you still have sickle cell? There's a lot of ambiguity that comes around that. And even for someone like me who is considered cured, I am phenotypically and genotypically like sickle cell trait. There is a possibility that you can still have the phenotype of your chronic illness. If you've gotten chronic pain for so long that it can't be reversed with the transplant or the gene therapy, is that a cure? And if you still don't have a kidney because you already had nephrotic issues while you had sickle cell, do you still consider that a cure? So there has to be a balance in that conversation, and taking into all these little things that compound into a patient's decision to go through this and that whole process. And oh, I didn't even have these on here. I'm so sorry. Well, I hope that makes sense. And lastly, I just want to reiterate that patient awareness, education, access to, and enthusiasm for gene therapy begins with clinician education, clinician awareness, and willingness to engage in and or refer for discussion about gene therapy. And clinician preparedness is very heavily influenced by the prioritization, the institutional prioritization of patients with sickle cell disease. And so I think a few steps that really helped in my process was having conversations with my clinician. Dr. Walters is my transplant doctor. And over several visits, he would bring up the topics that were really important for us to think about: the risks, the benefits. I could die. And everything was very balanced. He gave me the opportunity to ask questions. He gave me the opportunity to change my mind. And that is really important. And I think every patient with sickle cell disease should have access to a knowledgeable provider who can give them a consult with all their therapeutic and curative options. I also think that, I already mentioned that. So yes, in the meantime, there are current resources that I use and other patients with sickle cell can use to help educate themselves about gene therapy. There's a Democratizing Education for Sickle Cell Disease Gene Therapy project. There are community-based organizations like Cayenne Wellness, Access Advocacy, Sickle Cell 101, ASH, and they all help to spread information to patients in a digestible way, and also utilize things like YouTube and Instagram and social media to help reach the audience and gather what patients actually want to know about. So I think this sentence really captures everything that we talked about today, and that's improving health equity requires a holistic approach. Change is needed everywhere, from the laboratory to the bedside to the boardroom, and how payers pay for care to health policy changes. This is not a one size fits all, and I hope you enjoy this talk. Thank you.
Okay, thank you all so much. In the interest of time, I will be brief with questions and then we'll move to the audience, but I'll start sequentially, so with Marsha. And this is leading into the need for something like an affordability task force that Manar mentioned. With the graph that you showed on the disproportionate funding that shows the greatest disparity around foundation funding in particular, I found that striking because it is often, these foundation fundings are often driven by family advocacy and a general structural empowerment that is clearly not as easily found within the sickle cell community. And so considering those structural barriers of financial to time, resources, to feeling socially empowered generally, I'm wondering what you would say about that tension between empowering communities so there could be more funding there versus focusing more on the structural barriers and the role that other players like the IGI can play in improving these models.
Okay, that's a really great question. And I would link us back to at UCSF, we're an anti-racist institution. And so the first topic is in that kind of initiative is, of course, recognizing that racism is at play. But then next, resources should be disseminated according to need. And you're right that historically, African Americans, Blacks and African Americans are not generating or able to generate that intergenerational wealth because of repeated ongoing barriers. And so allyship would be what I would say is the step that is needed to really make a difference. And that doesn't mean that, allies who have more wealth dictate how the population should move forward or what people should be doing, but rather should be working in partnership, should be listening to the community. There's a saying, nothing for us without us. And so the money should come to the community, but the community needs to have a voice. And it shouldn't be, again, that other populations are dictating to the Sickle community, what their priorities should be. It should be generated by that population. But allyship is critical because, as you said, the resources can be really limited.
Thank you. So moving to Manar then, and thinking more about the logistics of what is possible through the IGI task force. You described the enormous systemic cost pressures involved, and thinking about this from a health system level, how it might not just be up to an institute like the IGI, but there are all sorts of players here that need to be considered, from insurance companies to hospital repayments with the medications and so forth. And I'm wondering, beyond the regulatory burdens and the changes to reimbursement models that you mentioned, which are very critical and huge tasks, I'm wondering if you could elaborate on what a not-for-profit pharmaceutical company could really look like, given that it is operating within this broader health system.
Yeah, that's a great question. So initially, when we embarked on the affordability task force, we really were somewhat narrow in our scope, and we wanted to identify what the IGI specifically could do. And then once we got the experts into the room, we realized that really what's of interest is what is a roadmap for the field to move into a parallel track to for-profit and venture capital, that model, right? So we are still going to need that. There are still many, many disease indications where the patient population is just too high. The cost will be tremendous. No academic or nonprofit institution could ever remain sustainable on just philanthropic funding. But there is a subset of commercially not viable therapies or smaller patient population sizes where a nonprofit or a public benefit corporation could come in and really provide an alternative and sustainable model. And so I think, the task force came up with a lot of really interesting recommendations, but to kind of combine two aspects of it, I think pricing and organizational models, to answer your question, I think there are two key recommendations here. One, that the price of the final therapy should really be tethered to the pipeline rather than being focused on maximizing profit for shareholders or depending on really high cost capital investment. We're really talking about anchoring the price to the cost of goods, to the cost of labor, and then relying on lower cost capital like social venture capital or philanthropic donations. And so from an organizational perspective, an entity or a combined entity that could implement such a new pricing model would, based on the recommendations of the task force, really be a mixed model that at different stages of the development pipeline, you would have different entities. So the discovery and the preclinical work would be done by an academic institution like the IGI or UCSF. And then you would have an intermediary for phase 1-2 trials that is structured as a nonprofit pharmaceutical industry. So that would be tax exempt and could rely heavily on philanthropic funding and government grants, but does have restrictions on what it can do and how effective it can be. And so we would, the recommendation is to place the licensing and the IP, as well as any eventual FDA approval within the nonprofit. and then to have an independent public benefit corporation that is still governed by an overlapping governing board. So there's some legal complexity there, but essentially saying that the public benefit corporation has in its charter a set rule and set operating procedures and what it can and cannot do into the future, whether it can sell its assets or not. And so at that public benefit corporation, which is essentially a for-profit corporation that has the legal ability to not only focus on maximizing profit for shareholders, but also pursuing a public benefit, which is why it's called a public benefit corporation. And so the public benefit corporation would take on phase 3 trials, would take on manufacturing and distribution, all of which are expensive. And so and there are some examples of this kind of mixed model in the field. So if any of you know of Civica RX, it's kind of a forerunner for and really an example for this mixed model in the generic space. And so it hasn't really been tried in the brand name or the sort of innovative therapeutic space, but really the idea is that you could based on the pipeline and where the cost pressures are, you could have different entities and then leverage different types of funding models and investments there.
Excellent. And there is a forthcoming report, I believe, which will all.
Yes, we are
soon.
putting out the report any week now, hopefully. But it is undergoing external review, so we really want to make it robust and strengthen the report before putting it out.
Okay, so focusing now on what happens on the ground and in the clinic. Turning to you, Christelle, you're in such a unique position in occupying that space of both patient and aspiring clinician. And I'm wondering when you were talking about how providers can improve their expertise and sensitivity when they're talking with patients about the prospect of these experimental therapies and when they become standard of care, how can they be more realistic about discussing those risk-benefit calculations so that we're not just talking about the hype and playing on people's hope? And you described how Dr. Walters did a good job of laying that out for you. And so if you could just share a little bit about what that looks like in conversation.
Yeah, is this on? Yes, okay. That is such a good question, and I've mulled on it quite a bit. I wish I had like a straight answer, but I think I can talk about the things that went well and the things that didn't go well. I think what is really important, I think as a foundation, it's important to hear the patient for what they're saying as opposed to the numbers that are on the labs. I think that can really get in the way of seeing whether or not alternative therapy or curative therapy is necessary. And so on my labs, like my kidneys aren't that bad and my AVN is not that bad, but I am coming to you telling you that I am in pain every single day for the last year and I've had to drop out of medical school and I can't wash my own hair. That is, even though the numbers are saying that I'm all right and I am socially still moving forward in my life, my quality of life and my suffering and the things that I'm telling you, like that has an immense value in itself. And I think hearing that and taking that into consideration just as much as the numbers, I think is really important when having that conversation with your patient about whether or not this is something they should consider pursuing now or later, either one. The things that really went well with Dr. Walters is that he was very straightforward. There was no ulterior motive. There was no, I want you to do this or that. It was a very objective presentation of the information and with lots of room for me to interject my thoughts and to ask a lot of questions. And I think the thing that is really important is that I know where I stand. I know what I want, but I want to know that my clinician has my best interest in mind and is an expert in the area. And that is what Dr. Walter showed me. He showed me that he wanted what was best for me, that he would let me choose what I wanted, but would give his expertise. And on top of that, what I love the most is that he'll say he doesn't know when he doesn't know. And I don't expect a clinician to know everything because nobody knows everything. But he's very forthcoming about that. And I think that was a really big oversight when it came to having that conversation in other areas, is someone pretending they really, really know about, or not pretending, but they really do believe that it's right. But if they've checked a few more things or asked a few more people or for me, it's slightly different. And so, yeah, those are some key points.
Great. That humility is is really important, isn't it? Okay, so I will, in the couple of minutes we've got left between us before we open it up to the audience, do any of you have any questions you'd like to ask of each other or further comments? Or I can just keep asking questions. Okay. Well, Marsha, I'll go back to you. With the social determinants of health and the different components that you laid out, social and community context is very important, and that question of acceptance in terms of how these therapies might be received in different cultural contexts, I would say, is really critical. And I know you've done a lot of work in Ghana, and thinking about that that statistic you mentioned with the mortality rates in sub-Saharan Africa, 50 to 90% of children dying before the age of 5. This is a profound issue, but I'm wondering how the goals and the conversations around gene therapy for sickle cell disease translate to the Ghanaian context, given all the different social contexts.
Yeah, that's a great question. And this last October, I was in Ghana working with genetic counseling students, and Dr. Walters was on Zoom. And so we were presenting about issues related to the gene therapies. And they were striking in their concern about the fertility issues, like upfront. And again, not that other people aren't concerned about that, but they just really put out that this is going to be a problem. So, but then, we talked it through and talked about how, how that could be overcome and what could be done. And then they again were bringing up, well, since this is so many children aren't going to survive, then it creates that dilemma of the parent making the decision for the child to go ahead with it. So we had, just a great conversation, and we've done focus groups and surveys to understand people's perspectives. I think the another issue really is trust, just as it is with this country. So again, when the Sickle in Africa Consortium is really about empowering African scientists and clinicians, and Dr. Julie McConney sets forward, the guidelines for clinical care across the continent for sickle cell disease. And so, again, everyone's voice is at the table, and it's a partnership with NHLBI and NIH. And we listen to what the concerns are. And again, trust is an issue given a history of exploitation on the continent. So there's a lot of work to be done. And I think another model that was brought up, in Ghana is the public-private partnerships, as well as governmental. So, and with the patient voice in there all along. And so that there's a big sickle cell center being built in Ghana that is a partnership. That it couldn't be done just with the government of Ghana. It couldn't be done just with the people of Ghana. It had to include some infusion of funds as well from a biotech company. So these things are, how that's going on the continent.
Thank you. Yes, let's open it up to the general audience. Sandrine, do we have any questions from online?
Not yet. There's one comment on the Q&A that it's such a great panel, and thank you, and a special shout out to Cell for being such a great presenter on the dual experience. And I just want to echo, like, I think each of you could easily fill one or more grand rounds, so we'll sign you up later for next year. But this is really great. Any questions in the audience? I have one, a comment slash question, and when I say things like this, I have to always preface it that I grew up in a country with socialized medicine, so I come from that experience and lens, and I hear this issue of the enormous cost associated with drug development. But I often wonder, I love this idea of non-profit, not-for-profit pharmaceutical industries, because this is a naive view of the world. Occasionally looking at advertisements on TV for drugs, to me it seems that most drug companies are really trying to get a better drug or a drug for the large masses of worried wells that are going to make a lot of profit, and spend relatively little time to think about the markets like the sickle cell market, which is not a small market. And so what would it look like if the government actually had much more control, or some entity had much more control, the FDA had much more control over what drugs are actually being developed? Because a lot of the cost, I think, is driven by all the unsuccessful ones and by these non-inferiority trials that are really only done to see if they can become the biggest holder of that market that already has a perfectly fine drug that doesn't need to be competed with.
That's a very big question. And I could probably talk for 15 minutes to try and give you an answer. Yeah, I think there's I want to touch on a couple of things. So one, I'm actually from Europe as well. And I'm used to socialized healthcare, but what also ends up happening in Europe is that pharmaceutical companies that have developed their drugs in the US actually sometimes don't market their things in Europe because they cannot get the price that they want. So for example, Bluebird Bio actually pulled out of the European market on their beta thalassemia drug, and simply couldn't come to an agreement with the Europeans on a price. And so that means that for the time being, these patients just simply will not access it. And there certainly is, there is not only an incentive from for-profit companies to make money, but in many ways they have to. So for example, Zolgensma for spinal muscular atrophy from Novartis has been dosed in 2,500 patients so far, with about 300 or so pro bono around the world. And it's only made $2.8 billion so far, but Novartis acquired it from Avexis for $8.7 billion. So they have several years to go to actually make up that money. So as much as I like to say that for-profit companies are doing this all out of greed, and many things they do are, there is also a big component of this actually costs a lot of money. And it's hard to recoup that money if not everybody is willing to kind of pay up or pay into funds. That's one thing. The other thing is about FDA sort of directing which drugs are being worked on. I think that's difficult. I think a lot of it is NIH funding that goes to basic science, which then fuels discoveries. And so I personally think that should remain open as possible, right? Like or basic scientists and running after their own curiosities has developed things like CRISPR, right? Which we wouldn't have if FDA had said, you have to work on this disease. But that being said, FDA and Congress have passed legislation to incentivize, especially rare and orphan drugs. So there's the Orphan Drug Act, and there are things like priority review vouchers that FDA can give out to incentivize for-profit companies to work on things that are rare. The problem comes in when it's really extremely rare, like 10 or 15 patients a year in, let's say, all of North America, and then no for-profit company will go through the hassle of running manufacturing and clinical trials for such a small patient population. And I think there are some talks at FDA on how to enable academic institutions to do this better. I don't know if I answered all parts of your question, certainly not, but I think it's.
Do we have any last minute questions? I think we're nearly out of time.
Summary thoughts from the panel. There are.
I actually have
summary.
a question on the last point from Dr. Treadwell. So you'd mentioned a lot of concerns in Ghana and sub-Saharan Africa on fertility. Does it ever come up whether malaria, endemic malaria, and potentially removing that protective protection that is derived from having sickle cell as an issue for the population? Is that ever discussed, or is it scientifically just not a thing because you can never correct 100% of all cells?
Yeah, that's interesting that that hasn't come up. Again, the costs, out-of-pocket costs for basic healthcare, those types of things people have brought up, but that hasn't been mentioned. Interesting.
Okay, last minute thoughts.
It's a little bit close, but maybe.
Okay.
Panel.
Thank you all so much. Conversation can continue.
Thank you so much for being here. This was really great. And yeah, I'm going to invite all of you back.
And we'll have to do another bioethics piece crossover grand rounds again. This is great.
Yeah, I didn't get to